Okay, now it's 9 o'clock. So thanks everybody for logging in this morning. We are recording it, and I sent out a link to last week's recording, so that's also where this recording will go. It's actually on the SSARE webinar website on the, I guess it's the new UADA/UAEX website. So this morning, Dr. Eva Wray is going to talk to us about parasites in small ruminants. That's the #1 thing we deal with producers. So again, at the end, there's going to be a quick little poll for you to complete before you log off. So Dr. Wray, I'll let you take over.
Okay, so I was told, just a forewarning, I was told the last talk I gave on Zoom that I flail with my hands and it's distracting, so you might want to collapse the video feed on the right. So I am Eva Wray. I will talk to you today about the internal parasites of small ruminants. Like Chelsey said, is the #1 problem worldwide of small ruminants and camelids. So alpacas, llamas, things like that. They fall under this umbrella. I'm going to go through it kind of fast. Chelsey is going to distribute my presentation to you, so there's a lot of information here. I just didn't want to leave anything out, so if I go over something too quickly, just let me know if you need it further explained. Okay, hold on. Oh, okay, so today we're just going to talk about who the parasites are, talk a little bit about the drugs. Again, I'm going to go over the drug information fairly quickly. Talk a little bit about why parasite control is important and how we can mitigate parasites. So this graph shows the changes in the gastrointestinal tract, the individual organs, their weight after infection with parasites. So the abomasum is where the barber pole worm resides. And this increase is not only due to the mass of the worms, but all of the inflammation, the edema. These worms, the larvae invade the tissue. So you have a lot of inflammation and immunological things going on, which creates an increased size in the organs. Same with the large intestine, you have worms that invade the tissue. So you're going to get a lot of edema, inflammation, things like that on top of the worm populations. And if y'all have questions, I would rather you ask as I'm going over it, just so I don't lose track of what I'm talking about. This graph just shows this parasite pressure on the animals as it's influenced by the season. So the barber pole worm is a hot weather worm. So you have a huge bump in parasite pressure when the weather is warm. So typically it's through February till about October, but that can be extended if we have mild winters. The last few years we have had trouble with hymoncosis affecting animals in December and January, which we haven't traditionally had. So that's something you need to be aware of with your producers. If they are having signs of hymoncosis in the winter, it could be hymoncosis. So fecal egg counts are very important on tracking that. This graph just shows the normal levels of nematodes in our small ruminants in the south. Hymonchus contortus, you can have up to 50,000 adults, no problem. And in the winter, you can have up to 1 million of their larvae encysted in that abomasum. So you can have a really intense outbreak in the fall, or in the spring, I'm sorry. Trigastronulus colubriformis, we'll talk a little bit about this worm. It's kind of an ever-present worm. It doesn't cause a lot of pathology. We find it in almost every sample we look at in small ruminants. It kind of rides the coattails of hymonchus, and it can have a pretty healthy population in that animal. Pneumatic Nematodiris, these are very large worms. They're in the small intestine. We see a couple eggs pretty regularly in fecal egg counts, but they're typically concentrated on dairy operations or operations where it's wetter. Esophagastum, we're not even going to talk about this one today. This one is a large intestine worm. It can develop 5,000 worms for this, or 5,000 individuals for this parasite is significant, but it's typically not too much of a problem in small ruminants. EPG is eggs per gram. This is what our fecal egg count results are expressed in. 8,000 eggs per gram is no problem with small ruminants. We've seen animals with 25,000 eggs per gram pretty routinely. So 8,000 is a pretty conservative population of parasites. Very quickly, the typical life cycle for these worms. Now this is the typical life cycle. Each worm does have have little tangents and changes that they undergo, but this is the typical. So the adults are in the host animal, whichever organ they're in. They mate, the eggs are passed in the feces. In that fecal pellet, the eggs undergo a couple changes within that fecal pellet. So they are protected from the outside for the first two or three life stages that they undergo. Once they hit the infective stage, they leave the fecal pellet and migrate out onto the grass. That's more concentrated in the mornings and evenings, especially in the summer months, where the dew content is higher than the rest of the day. And coincidentally, that is when our animals are grazing the most in the hot summer months. These worms do require moisture to be able to move freely up and down the grasses. So you'll get a surge in parasitism infections after a heavy rainfall. Once these worms are migrating up and down the grass, the animals eat them, and then they're passed on, or then they move to the site, and then life cycle starts all over. Start to finish, this is about 3 weeks, 3, 4, 5 weeks, something like that. So it's very, very quick. You can develop really high populations on your pastures in a relatively short amount of time. The important worms, the nematodes, Hymonchus is number one. All of these others, I'll give you a little information about them, but Hymonchus, the barber pole worm, is the worm to worry about. As I said, Trigastronulus is kind of ever present. It doesn't do a whole lot of damage. Nematodiris can be significant, but it's concentrated on specific operations. The deer worm, the brain worm, I'm sure a lot of you that deal with small ruminant producers have had kind of an increase in the interest in this worm. It's can be detrimental, and we'll talk a little bit about that. Again, it's not on every operation, and it's not every animal, so we'll talk a little bit about that. Other important helminths, again, Hymonchus is the number one. Tapeworms, the sheep tapeworm is pretty common. We see it in probably 80% of our samples. It really doesn't do a whole lot of damage to the animals, so it's unsightly more than anything. Then we'll talk these two other tapeworms. Taenia ovis is spread by dogs. It can cause a problem in your sheep, especially if you're selling the meat. It'll cause causing them to condemn the meat and the organs at slaughter. Echinococcus is a tapeworm that humans can get, and it can be important. It's not common and it's not rare. It does happen, but it's not as common as all of these others. The liver fluke, this fluke is also, this worm is concentrated on certain operations that have standing water in their pastures. Typically not a problem. I've never seen a liver fluke in the decade that I've been here, but the operations that do have liver flukes, everybody's infected, and it can be detrimental. So we'll talk a little bit about that. So Hymonchus, barber pole, these are female worms here. They look like candy canes or the barber poles. So the white are the eggs and the ovaries. The red is all the blood that they suck, and they're very substantial worms. They're very large. So 60,000, 50,000 of these all sucking blood can kill your animal very quickly. As I said, this worm is in the abomasum, which is the primary site of the start of protein digestion. Very important organ. And so if you have a lot of protein loss through blood, then you can, and compromising this organ, you can have an animal go down very quickly. They are voraciously hematophagic. They suck a ton of blood. An acute infection with Hymonchus can take nearly a quarter of an animal's blood in a single day. So we've had issues with this with our study animals. We will, in the morning, we'll go out, they look fine, they're eating, and then by 3 o'clock that in the afternoon, they're dead. So they can go down very, very quickly with this worm. They are a hot weather worm. They do physically invade the abomasum, the tissue during the winter, so they can overwinter until environmental conditions are efficient or correct for them to continue their life cycle. There are two phenomena associated with Hymonchus. It's called spring rise and periparturient rise, or post-pregnancy rise. So in the winter, these worms are encysted in the abomasal wall, and when in the springtime comes, they get environmental cues. We're not really sure what those cues exactly are. They could be hormonal, not really sure what they are. And this affects both males and female host animals. What will happen is all of these, a huge portion of these encysted larvae will all come out, emerge at once, and then turn around and start feeding on the organ. So not only are you having blood loss from all of these larvae feeding, but also tissue damage from the cysts that were broken open and now are exposed, and for potential secondary bacteria, illnesses, things like that. So female host animals, along with spring rise, also undergo a post-pregnancy rise. So once they start lactating and they get lactational hormones flowing through their body, they'll have another wave of larvae emerge from their cysts and turn around and start feeding. So female animals, goats and sheep, in the springtime, they are at the highest risk of dying because they're, stressed from pregnancy. They may have post-pregnancy issues, along with all of this huge population of parasites feeding on them and stealing further nutrition. This graph just exemplifies spring rise, periparturient rise, and then the blue line is a ewe that has a stillborn lamb. So spring rise is the black line. This happens again, males and females. The spring hits, you have a bump in fecal egg count, a high population, and it starts to fade off in the winter. Periparturient rise, again, you always have to keep in mind spring rise and periparturient rise are combined in female animals. So not only do the females have this spring bump, but they also have this huge periparturient bump, and so they can become overwhelmed really quickly. Something interesting that we've seen is a ewe that is pregnant, she's giving pregnancy cues, starts to have an increase in her fecal egg count, and then once she has a stillborn, those cues are no longer intact, and her egg count drops back down to the spring rise levels. This is a graph that shows the periparturient rise in different numbers of offspring. So the periparturient rise is directly correlated with how many lambs or kids are put out on pasture. So you have 0 lambs, just the spring rise, you have a small bump for 1 lamb, and then it goes up for every lamb that you add, lamb or kid that you add, you do see a significant bump in that fecal egg count. So you have all of these lambs out on pasture, and these parasites know that they are naive animals and that they can get an infection established in them, so they bump up their numbers in order to perpetuate themselves. Hymonchus is extremely reproductive. A single female can produce about 5,000 eggs in a single day. And again, if you have 20,000 females, that's a significant population. They're very, very pathogenic. This could be due to humans trying to intervene in the in this worm's life cycle. It has been shown that drug intervention and human intervention has created more pathogenic worms. So not only is Hymonchus resistant to just about everything, we have created a more powerful, hungry, pathogenic worm. So we kind of backed ourselves into a corner. Patency, which they can live up to 8 months. So a single worm can live for an entire grazing season. So that's significant. It has displayed resistance to all classes of drugs, especially benzomidazoles and ivermectins. Ivermectins are the ivermectins. They've been grossly overused in the years with all producers, so that they've developed resistance to that. And the most important aspect of small ruminant production. Signs are pretty common, or pretty typical. Sudden death typically is the first clue that your producer will see that they have a worm problem. They'll just start dying. They'll have a heavy death rate. Bottle jaw is another one. I'll show you some pictures. It kind of looks like a parrot mouth. And then pale mucous membranes. You can see this through your Fromaggio scoring. And then all of these other conditions, all these other signs can be confused with other things, but if you have one or more of these other signs, then you probably have Hymonchus. I guess I don't have pictures of bottle gel. Trigastronulus, again, not a very pathogenic worm. It's in the small intestine. It has low reproduction, low pathogenesis. It's susceptible to most anthelmintics, so very easily to get rid of, but it's ever present. Nematodiris. This Mustang worm, this is the adult male here. These are the infective larvae. It goes from infective larvae to adult in a few weeks, and all of this mass, all of this gain is from your animal. They're taking tissue and blood away from your animal, so they can be significant. Again, kind of confined to certain operations, operations that have pastures that are for new animals that, every year. They do, let's see, cold weather worm. So it's the opposite of Hymonchus. These worms thrive in the winter. They are present in warmer weather, but they do much better in the colder weathers. They persist on pastures for about 2 years in the absence of animals. This is due because the first 3 stages are all confined in their eggshell, so they have not only the protection of the fecal pellet, but also the several layers of that egg that protect them from the environment. So they can persist on pastures for a long time, longer than any other worm. So it can be significant. And again, operations that have this have traditionally had a problem with it for many years because of this persistence on the pasture. The sign that you have, Nematodiris, is a non-bloody diarrhea, and it is a projectile. I've seen cattle with Nematodiris project their feces 5 feet away. It's pretty impressive. So if you have a non-bloody diarrhea and they're sunken up and dehydrated, you might want to test for Nematodiris. Typically are limited to younger animals, although if you have a heavy Hymonchus population in your animals, it can weaken the immune system enough that even healthy young or older animals can get this infection. again, it can become significant. The adult worms will trigger a thigmokinetic response in the host. So what they do, and this is what causes the intense diarrhea. So they will physically wind themselves around the villi in the small intestine, and they'll strangle it to the point where the body will reject that villus, and then they'll move on to another villi. So they're kind of going through and causing a huge rejection of the villi, which can cause diarrhea. They are the dose-limiting worm for dose-limiting nematodes for ivermectins. Let's see. Chelsey asked what dewormer is labeled for Nematodiris. I'm not sure. I know benzomidazoles work against them. Ivermectins, they do show resistance to ivermectin. So dose-limiting nematode, this means that they require the highest dose of the ivermectins to kill them. I think it's something like 2 or 3 times what all of the other nematodes need to kill it. So you have to give it a big dose to kill this worm if you're using ivermectins. Benzomidazoles, your white wormers, are still efficacious against this worm. So if you do have Nematodiris, I would recommend, one of the only times I would recommend benzomidazoles in small ruminants, but it does work. Okay. Okay, so the deer worm, the brain worm, the meningeal worm, it's not, our sheep and goats and camelids are not natural hosts for this worm. So when they do get into the wrong host, that isn't a white-tailed deer, they develop all of these neurological problems. They stumble, they fall, they tilt their head. So if you see any neurological stuff in with your producers, I would test for this worm. All gastropods except cattle get this, or act as accidental hosts. So moose, elk, goats, sheep, everything except cattle. They don't really know why cattle, they don't affect cattle, but they don't, so that's a good thing. The life cycle does require snails and slugs. So to prevent that, you can try to prevent snails and slugs on your place, but or block it off so deer can't get on your place, but it's very difficult. Drugs are very limited to what can be used against these because they are beneath the blood-brain barrier, and not a lot actively crosses the blood-brain barrier, so the dewormers can't even get to the worm. Ivermectin is a drug that's commonly used for this worm, but it does not cross the blood-brain barrier. So it might be effective against the migrating larvae, but it does nothing against the adults that are located on the brain. Safeguard does penetrate the barrier, but it has very limited activity. We're very limited to what we can do with this worm. Steroids and anti-seizure drugs are typically given with the anthelmintics to reduce inflammation, keep the subsequent pathology quieted, so the animals can recover. And with this worm, some animals recover, some don't, both with and without treatment. So it's kind of a crapshoot as to who's going to live and if your drugs are going to work. Prevention is very difficult. Like I said, you can reduce or eliminate the deer, snails, and slugs. But that if anyone is successful in doing this, please let me know so I can pass it on to my producers as well. So just a few gross pictures. They're very, up here is the brains here. This is the skull. It's a very large worm. Here down at the bottom left, it's very large, so you can see how it can cause a lot of neurological issues. Clinical signs, again, anything neurological, if they can't stand up, they sit like a dog. Here's some pictures. If you see anything like this and they seem fine otherwise, it's just neurological, it could be an indicator that you have this worm. Tapeworms, again, we're just going to very briefly go over this. The adult is in the small intestine, eggs shed in the feces. Those eggs are consumed by a free-living mite. That mite is ingested by the animal and the life cycle starts over. It's not
very pathological in small ruminants. You can get enough impaction, a big enough population that you can cause intestinal impaction. It's very rare. The adults can get 10 feet long, but again, and tapeworms do not feed directly on the animals. They only consume the intestinal contents. So they bathe in their food and absorb the intestinal contents through their skin, their tegument. So they're not feeding on your animal. They do steal nutrients, and they do cause, where they're glued to the small intestine, they do disrupt or compromise the absorption and the breaking down of nutrients. But typically, they're just unsightly. People don't like to see the white perglottids in the feces. You'll see these. This is a very common sight. White wormers, benzomidazoles do a really good job against tapeworms. Unfortunately, they don't do much of anything for any other worms except Nematodiris. So, but if your producer has tapeworms, just tell them you give them some safeguard. It's no problem. Flukes, just very quickly, the adults are in the liver. The eggs are passed in the feces into direct water. The requirement of this worm is the feces has to land in water, because the stage that exits the fecal pellets swim to find these mud snails that are in the water. So water is a vital part of this life cycle. We typically don't have problems. Like I said, I've never run into flukes up in Northwest Arkansas, but once you start getting to the River Valley, East Arkansas, lower lying areas, river bottom pastures, you could start running into flukes. Once they undergo a couple changes in the snail, they exit out, swim to the blades of the grass, and they will swim up to the top of the blade and encysst themselves. So they will create a little crust, and there they sit until they are ingested by the animal. So once they're in this cyst, they're extremely environmentally resistant, so they can persist for a while. Once that animal eats it, it moves to the liver, and the cycle starts over. So once the fluke gets into the host animal, they move to the liver, and they undergo a migration throughout the liver until they finally settle in the bile ducts. With sheep and cattle, this is typically a week or two of migration, and then they move to the bile ducts. With goats, they never stop migrating. So they just for some reason, a goat is a somewhat unnatural host for them, and they never get the cues to continue on their life cycle. So they migrate and migrate and migrate until the liver is just jelly. So extremely detrimental to goats. They're very big, very, very big flukes. They're hermaphroditic, so no males and females, just one creature. Very gross. Let's get it. Oh, I thought I put some liver pictures in here. So, oh, I did. Yeah, I think I've said all this. You can use a fecal egg count to diagnose, and flukicides to treat. Flukicides are always combined with another anthelmintic, another drug. And they typically have plus, so it'd be Ivermectin plus, Ivermectin plus. That plus indicates that there's a flukicide in that to treat flukes. Acute fascioliasis is not very common. This happens after we've had an extreme drought. The snails have gone under the mud after you get a heavy rainfall. All of these snails come out of the mud, so you get a big surge of infectious stages on the grass after a rain. So and all of this huge population moving into the animal, infecting the animal, they're all migrating at once, and it can cause a liver hemorrhage and can cause death in a week. And all of these migrations are compounded by the presence of Clostridium. It's called black disease. All of these, the migration through the liver causes wounds, which then the Clostridium can take hold, and that's very common.
Clostridium is a big driver of the pathology of this worm. Chronic fasciolisis, this is the most common form. It's just long-term infection. You get pipe stem liver, which is just hardened bile ducts. I think I have pictures in a little bit. Disruption of the liver, malfunction of the liver. You get anemia and jaundice, progressive psoriasis or cirrhosis, and progression. So you get a progressive condition loss. So typically when I have producers that contact me and I think they have liver flukes, typically what they've told me is they've treated with antibiotics, they've treated with every dewormer they can find, they've done all of these things, they've moved them off of the pastures, they put them in barns, but they still keep getting worse. And if I ask them if they have standing water on their pastures, and if they say yes, then they have liver flukes. That's kind of how I diagnose it. Again, don't come in contact with it much, but so I kind of say if everything else has failed and they have water in their pastures, regardless of how much water, and the animals are still getting worse, it could be liver flukes. To control it, you treat the infections, the adult infections. I'm not aware of any drug that actually kills the fluke larvae. So you're just treating the adult infections, and so you might have to have subsequent treatments every couple of weeks in order to catch all of those larvae that have grown into adults and kill them once they are adults. Rabbits and deer kind of drive this life cycle. They also get this parasite. So again, if you can fence them off and keep them off your place, that would be best, or that would be good. But the best way to do it is to eliminate or fence off the standing water, even with electric fence or panels or something, just to keep those animals off of that water until it's completely dried up. And if they can forever keep them out of that standing water, off of that patch, then that would be ideal. But we have a lot of producers that don't have a lot of land to spare for their animals, so you just have to work with each one individual. So this is pipe stem liver up here on the left. This entire bile duct, all of these bile ducts are completely non-functional. So this and flukes will cause rejection of the liver at slaughter, so it can be important. This is hemorrhaging. This is acute on the right. It's acute fasciolisis, and then down here you can see the X's are the flukes. So you can see they're pretty big, pretty large. So the drugs, the ones that are highlighted, that are bold, are the drugs that I would recommend. Levamisole is probably my go-to recommendation, just because it's been around since the 40s, but it is somewhat toxic, so folks quit using it for a while. So it is still efficacious against Haemonchus, although we are seeing a drive in resistance because people are moving to this drug. It has a limited spectrum of activity. It doesn't get migrating larvae or encysted larvae. It requires suspension in water. I believe it only comes as prohibit now. In the past, it was Levazole and Tremazole. I'm not sure these products are available anymore, but prohibit comes in a powder form, a packet, and then you suspend it in so much water. It's very important that you tell your producers to get these conditions correct, get the correct amount of water and get the correct amount of drug into that animal, because it is toxic. It's a neurotoxin, and they can end up killing half of their animals or all of their animals if they do it wrong. It does help address microcyclic lactone and benzomidazole resistance, because it's a completely separate class, a different mechanism of action. So it does address resistance problems. In the past, we've had trouble getting a hold of this drug. I don't think that's the problem anymore. I think you can just go to the co-op and get. The next drug that I will recommend is Cydectin, which is a subclass of a macrocyclic lactone. It only comes in an oral formulation. There are macrocyclic lactone resistant strains of Haemonchus, so If you have, or if your producers have worms on their place that are resistant to ivermectin, Cydectin resistance will soon follow, just because they are in the same umbrella class of compound. Moxidectin does typically do better than the ivermectins for efficacy. This is due to it getting caught up in the fat. It lingers a little bit longer and it's not flushed out of the system quickly, so it does have a little bit of residual action usually. But again, if you have resistance on your place or your producers have resistance to ivermectins, moxidectin will follow shortly. Sulfonamide, chlorcelon, this is the adult fluticide. Again, it's combined with typically ivermectins, but in a plus formulation. It'll say plus or something like that, and that will indicate that it does have chlorcelon in it. Benzomidazoles, I do not like recommending white wormers to my producers. Hymonchus has, that was one of the first drugs that it showed resistance to back in the 80s. As soon as this came out, Hymonchus started showing resistance to it. So, and there are resistant strains of Trigastronulus as well. So you have to be careful with this recommendation. With that, and also valbazen does cause early term abortions in small ruminants. So if they give albendazole or valbazen to their animals and they are pregnant, they will abort everybody. So it's routinely used overseas as an abortion agent. Although valbazen is indicated for adult flukes. So albendazole and chlorcelon are the only two drugs that get adult flukes. So just, I try to stay away from benzomidazoles. And again, and white wormers do help kill tapeworms. But again, I try to steer my producers away from benzomidazoles because they're not very efficacious, and because if they're not careful in their application, they could abort their animals. Ivermectins, just very quickly, the ivermectins, they don't really work for any of the worms in small ruminants, just due to the overuse and the generic preparations of Ivomec. Generics just don't work as well as Pioneer, and it kind of they kind of drove the resistance problem. I never ever recommend ivermectins to my producers ever. Just to save them money and to save their animals. This is a table with all of the approved drugs in sheep and goats. As you can see, there aren't very many, nearly no drugs. So levamisole is not approved in goats. Moxidectin is not approved in goats, so you have to use it off-label. Nothing on here for alpacas or llamas. So a lot of this stuff is off-label. So just keep that in mind and maybe let your producers know that. So what makes these parasites successful? We talked about a Hymonchus female can produce 5,000 eggs in a single day. If you have 100, just a super conservative figure of 100 female nematodes in one animal, or okay, let's see, in one animal, you get 500,000 eggs a day on pasture from one animal. If you compound that to 10 ewes, you get 5 million eggs on pasture a day, which results in about 50,000 new infective larvae on your pasture every single day, with an extremely conservative number of adult female nematodes in that animal. So our producers that barn their animals at night or lock them, they don't allow them to graze, you can get an extremely high concentration of infective larvae on your lots very, very quickly. So that's something to keep in mind. Survivability is another reason for success. We talked about the fecal pellet protection. The first couple stages are physically inside of that fecal pellet, protected from the environment. On top of that, you have their the nematode eggs that they have, the helminth eggs, have several layers to them. So again, they have shell protection. Once the infective larvae move out onto the pasture. Once they become an infective larvae, they have sort of a coat of arms around them. They protect them. They're ensheathed in this. They don't feed at this point, so that it's just all about protection. And they can persist for a long time on pasture. They're resistant to weather conditions. When the weather, for Hymonchus, when the weather starts getting really hot, they will burrow into the ground and stay there to try to find some moisture and stay there until the conditions become right. So very, very, very persistent. They're very fast and they react to light very quickly. So they're hydrophilic. They love water. High dew content. You can bet that you have a high concentration of parasites on your grass at that point. Anytime we have rain, several days after it rains, you're going to have a higher incidence of infection on your place. They're negatively geotropic. You don't need to know that. They move against gravity, so they move up the blades of grass. Inside the animal, they survive. They're very good at surviving. They have the seasonal arrestments, like I said. So you have hot weather worms and warm weather worms. When the environmental conditions are not optimal for the perpetuation of their life cycle, they insist inside that animal and just wait out the bad weather. This also is true of intraburden arrestments. So if the worm senses that it is taking, this is especially true with Hymonchus, although they typically don't catch it in time. But if the population senses that they're taking too much from that animal and they're going too quickly, they will halt all of their reproduction, their egg laying, their molting within the animal. Everything halts. Their feeding, they'll stop feeding and let that animal kind of try to bounce back, because they don't want to kill that animal, because if the animal dies, then they die. So their end goal isn't to kill the animal, it's to create a balance within it. But humans have intervened so much that we've created a super worm in a sense, and they take so much so quickly that sometimes they can't regulate themselves in time. But that is a tool that they use to survive. They can dodge the host immune system very easily. So gastrointestinal worms are already outside of the immune system. So if you think about it, from your mouth to your anus is technically outside of your body. So these worms are not even, it's inside of your body, but outside of your body, if that makes sense. So these gastrointestinal worms, unless they invade the tissue, they're outside of the purview of your immune system. So they're able to dodge it that way. And when they do invade the tissue, they wall themselves off in sort of a calcification, so the immune system can't get to them that way. So they're very good at that. They can develop resistance to a drug. Hymonchus can develop resistance to a drug in a couple generations, and a generation is about 3-4 weeks. So in 2 months' time, a drug can be worthless on a place if you use it too much. And I'm not going to go into the details of how they do that. They're able to change the way the drugs affect them. So just for example, one of the many ways they do this, they change the attachment sites of those drugs. For the next generation, the survivors of a drug treatment, their progeny will have, sometimes they'll have fewer attachment sites for that particular drug, whatever chemical was used. They will have more trash pumps in a sense, so they will be able to drag that drug in their body and pump it out before it's effective. They'll have 10 times the amounts of waste pumps that their predecessors had. So in one generation, they can change the way a drug affects their progeny. And the biggest part of their survivability is they stay non-clinical. Most of our food animals, most of our animals have parasitisms, but these parasites have struck a balance with these host animals, and so they stay non-clinical. And the producer has, if they're not monitoring it by fecal egg counts or Fibonacci scores or things like that, they have no idea that their animals are infected. So non-clinical parasitisms are a big driver of their success. The mistakes at the farm level, number one is administering the wrong dose. Most folks don't have scales. Most folks do not use a weight tape to weigh their animals. It's very hard to eyeball a sheep with its winter wool on, if it has, or if it's shedding. It's hard to determine the weight. And some of these drugs are yes, Sarah, I'll talk about copper boluses in just a little bit. But yes, so really encourage your producers to get a weight tape. They're way more accurate than eyeballing, and especially if you're recommending your producers use levamisole, which again is toxic. They don't want to be giving their animals 3 times the amount of drug that they need and kill that animal. Now, one thing that should be said at this point, goats have a much more efficient liver than sheep have, and camels, the camelids. So you have to give goats 1.5 times the amount of drug that you would give sheep, because their liver, they're used to eating toxic plants and things like that. So they have a very efficient liver. It kicks out toxins very quickly, and they kick out the drugs very quickly. So you have to use a higher dose for goats. Again, it's hard to determine what the dosage is if you're just eyeballing the animals. So wrong dosage rate, treating at the wrong time of year. Again, if you're after Hymonchus, treating in the winter, unless you've confirmed an infection with fecal egg counts, treating in the winter, you're just wasting your money. Treating too much. So Refugia is the population on an parasite population on an operation that has not been exposed to a particular chemical. So you want to have a wide Refugia, meaning that you want as many parasites on your operation as possible that have not been affected by a chemical. That way they are susceptible, their genetics are susceptible when the drug treatment is applied. And this happens when you cut back on your treatment. You don't want to blanket treat. You want to just spot treat the animals that are showing pathology by confirming that with fecal egg counts and FOMACHA scores. So you want to try to never expose your parasite populations to a chemical. So when you do use it, it works. Too much reliance on drugs. Drugs are not the answer anymore, especially in small ruminants and camelids. The Hymonchus is just too resistant to everything. So I would encourage you to encourage your producers to move towards non-chemical approaches, management approaches, nutrition approaches, things like that, and move away from relying on drugs, because if they rely on drugs, they will get in trouble very quickly. Drug problems, selection for resistance. We've talked about this. You want to expose your parasite populations as little as possible to drugs, so they don't develop the resistant genes and they're not creating progeny that have resistant genes. You want everything to be susceptible. So again, use drugs as little as possible. Pioneer versus generic. This is more relevant in cattle, because again, ivermectin doesn't work for anything in sheep and goats, or I wouldn't recommend it to my producers. In cattle, ivermectin has probably 200 generic formulations. Study after study after study that we've done, generic does not do as well as Pioneer. It can be in the same bottle, the same manufacturer, the same facilities, everything. The generics don't work as well for some reason, and we're not really sure why, but it has been shown in all of our research that we've done. We always throw generic in, it just doesn't do as well. So all of you that particularly cattle producers, I know this is sheep talk, but I would tell them to use Pioneer, just to save them money and heartache in the end. Stick with the Pioneer products. Product storage is a big one, especially in the summer months. producers will go treat their animals and then throw the drug on the dashboard of the truck or in the back seat of the pickup or in a hot barn or put it in a freezer. I've had a producer do that, and all of that will degrade the chemical and render it useless for the next time you use it. So store the product correctly. Sunlight breaks down the chemicals. So if you leave it on your dashboard and it's not in a light proof container, you will get degradation of that product. So product storage is big. Mitigation through management practices. I'll explain all of these. Fecal egg counts are probably the most important thing with small ruminants. It allows you to track your infections, identify problem animals before they become a problem and you're fighting for their life. You can follow the egg counts as they're coming up and treat accordingly. Once it gets a certain threshold, it will help produce, it'll decrease the kill rate, the death rate on producers, and save them money as far as identifying the animals that have all the parasites and getting them off their place. Fecal egg count reduction tests. Am I going, let me know if I'm going over too long, Chelsey. Fecal egg count reduction tests. This is a way to test your dewormers, if your dewormers are working through conducting fecal egg counts. I recommend doing a fecal egg count reduction test every single time you use a drug, because again, you can get resistance with one generation of worms. So if you're giving a drug and you're not testing if it's working, your producer could be wasting hundreds and hundreds of dollars on a drug that doesn't work. So that's important. Smart drenching is just a methodical checklist for treating, and we'll go over that. And then FOMACHA scoring, I'm sure you're all familiar with that. We'll talk a little bit about rotational grazing. I'm sure you're all familiar with it. Alternate species grazing, which is, sheep and goats, or sheep and cattle, or sheep and horses, things like that, grazing them behind one another. Copper oxide wire particles, very good tool. We'll talk a little bit about that. Condensed tannins, forages that create that have tannins in them do have nematocidal effects. Dung beetles, we'll talk about them for pasture health, very, very essential. And then Duddingtonia flagrans, this is bioworma. We'll talk a little bit about that. So non-chemical approaches as far as nutrition and management, increasing their protein intake. As I said, Hymonchus affects the abomasum, which is the protein digestion site. If you increase their protein, you're kind of replacing what those worms are taking through the blood. Increasing their minerals, it's very important to have free choice loose minerals. That way, again, they're able to replace all of that stuff that the worms are taking. Reducing stress. A stressed animal will have 10 times the worms that a non-stressed animal will have. I know it's hard to do with small ruminants because they're scared of everything, but it's very important. Overstocking. My official answer on diatomaceous earth is it is worthless. I'll say that to you because you're agents. It doesn't work. We've done my bosses have done studies where they've given so much diatomaceous earth in the feed that the feed turned chalky. The fecal pellets were white, and they were still loaded with nematode eggs. So it's good for external parasites on poultry. So giving the birds a dust bath, that really helps with mites and ticks and things like that. But as far as internal parasites go, tell them to save their money. That's, yeah. Preventing overstocking. Overstocking can lead to stress. And if you have a limited amount of land and you have too many animals on it, the parasite population, the infective parasite population would be astounding. So overstocking is a biggie. Repairing water tank leaks. This creates, so all the animals are coming to the water tank and defecating and all of this species, and it's wet and moisture and perfect conditions for parasites. So you want to repair these tank leaks that are driving this wetness and moisture and this concentration of worms. So fecal egg counts, you have a McMaster's technique and a direct flotation. In the lab, we do direct flotations. We use a centrifuge, and it's a little more involved than a McMaster's technique. McMaster's is what I teach my producers. Chelsey teaches a lot of her producers this as well. It's all it takes is a slide. It doesn't have centrifugation. It's very, very good to use in a field. Really all you would need expensive wise for expenses is a microscope, and you can get one of those on Amazon for $40 now. That would do just fine. So the two techniques, and hopefully Chelsey and I will be able to get back out and do McMaster's fecal egg count clinics, teaching our producers how to do that. You should always conduct fecal egg counts before administering a drug. If an animal doesn't have enough worms to treat for, you shouldn't treat that animal. You want to strike a balance in your animals. You want your animals to be able to hold X amount of worms and still be fat and happy and healthy and have babies and produce milk and all of these things and grow like they should. So you want to strike a balance. So fecal egg counts will tell you if these animals have reached the threshold, the treatment threshold. Otherwise, you don't want to treat them. Blanket treatments, I know it's been recommended in the past. Do not do blanket treatments anymore. This will just drive the resistance problems on operations, and it'll get your producers in trouble very, very, very quickly. What they need to do is identify the animals that need treatment through fecal egg counts and FAMACHA, and then just treat those singular animals. Fecal egg counts will reduce the incidence of resistance on operations. We kind of touched on this. Being able to track whether your drugs are working or if they've failed is essential. Being able to find out which animals have the most worms. So 20% of your animals on any given operation will harbor 80% of your parasites. And this is true with any homogenized group of animals. So this is true with cattle, with wild deer populations. 20% of those animals will have the majority of those worms. So through fecal egg counts, you can identify those 20% carriers and get them off of your place, because you don't want to pass those genetics on to your animals, which will result in parasite problems in the future. I've had, for example, my mom had sheep. About 8 years ago, she just had a heavy, just couldn't, no drug worked. She had a heavy death loss, and we started doing fecal egg counts to try to identify it. We found those 20% that were left. Most of the 20% had died due to their worm populations. We got them off her place, and now she hasn't dewormed her sheep. I think she dewormed one lamb last year, and hasn't done it because she got most of those parasites off of her place. And the remaining 80% of the animals were somewhat resilient to those worms. So very, very good tool, or very good thing that can be learned from fecal egg counts. And you should conduct fecal egg counts as frequently as possible. If I had sheep, I would do it maybe once a month or once every other month, especially in the summer months, in order to be able to accurately track those infections and cut off a problem before it becomes a problem. Fecal egg count reduction tests, again, just used to determine the resistance status of a given drug on a given farm. The guidelines, just very briefly, pull a fecal collection before you give a treatment, either day 0 or day -1. You don't want to be too far ahead. Treat that animal with a single drug, and then conduct a fecal egg count 2 weeks after that treatment on that same animal. And then you do a small calculation. If y'all need the formula, it's very easy. I can email that to y'all. And it's very accurate, and very good tool. Again, I recommend doing this with every time a drug is used. So things to consider: only use animals with an appropriate fecal egg count. Again, if you have animals with 3 eggs per gram or even 100 eggs per gram in sheep and goats, you don't want to treat those animals because it will be pointless and will drive resistance on your place. This one, I don't know why I run into this problem. At the end of the 2 weeks, pull the sample from the same animal that you used that you did on day 0. Otherwise, you won't be able to work in the formula. So that, be sure your producers know that. 28-day post-treatment fecal egg count can be helpful. It can help identify if those drugs knocked out the egg production or if they actually killed the worms. They had cidal effects, so that's also important. So follow-up, as many fecal egg count follow-ups as you can get in would be helpful. In the textbook, anything under 95% efficacious is resistant status. In reality, with sheep and goats, if you have a drug that is still working 70, 75, 80% efficacy, that's a good drug. 95%, I don't know of a single drug in any animal out there that has a 95% efficacious rate. So, but in the textbook, that's what it says, or in the guidelines, WAVP guidelines, that's what it says. So, but in reality, if you have a drug that's killing 75% of the worms, then that's good. You want to assess all your dewormers and keep thorough records. Smart drenching, we talked about this. You want to weigh your animals to get a proper dosage. Weight tapes are better than eyeballing it. Yes, I'm going to fly through these actually. Sorry, I always go over. Let's see. Know the life cycle, know what worms you're after. Record your deworming history, practice good biosecurity. You don't tell your producers never get cell barn animals. Never get cell barn animals. That's where we get our study animals. You want to use fecal egg count reduction tests to learn the resistance status. Use drugs till they're ineffective. You don't want to rotate dewormers. Practice good pasture management. FAMACHA scores, FAMACHA used to assess the blood loss, only associated with Hymonchus. It's a good tool, but you shouldn't treat both. This is the card. I'm sure you've all seen the card. Alternate species grazing. It's just rotating one species after another. Cattle are really good about cleaning up sheep parasites, horses, vice versa. If you have donkeys with your sheep, that's good. They act as dead-end hosts to those other parasites. Rotational grazing. Rotational grazing will allow you to control the height, the grazing height of your pastures. Those parasites only travel up the blades of grass 3 to 4 or 5 inches. So if you keep your pastures as high and lush as possible, you reduce the amount of interaction your hosts have with the parasites. Copper oxide wire particles. This is copper sulfate, not copper, or copper oxide, not copper sulfate, so it will not kill your animals, kill your sheep. This is only used on the existing adult infections. It doesn't do anything on subsequent reinfections, but this will allow your animals, especially animals that have just had babies, it will allow them a major kill off of the adults and kind of a reprieve in order to bounce back a little bit. Condensed tannins are forages that directly affect GI parasites. They damage the cuticle and the digestive tissues of the parasite. They do have a bitter taste, so you have to kind of train your animals to, and it's readily done, but you have to kind of train your animals to eat it, and goats are more tolerant. This is just a few examples. Dung beetles. Dung beetles are very good for your pasture health. They help break up the life cycle of the parasites. They either physically eat or remove the feces from the pasture, carrying those parasites along with them. And as an added bonus, they're also killing off horn and face flap populations of the larvae that are in those fecal pads. Ivermectins do kill dung beetles. Ivermectin kills dung beetles. Cydectin doesn't, but that's why we had such a kill off, is because ivermectin became readily available. Bioworma is a biological control. It's a fungus that kills nematodes. Fungal spores are fed as a top dress daily. What they do is they kill off the parasites on the pasture before they become infective. In the research has been shown to be very promising, although the application is laborious. You have to feed it every day. You have to get the right amount every day. It's very expensive, but it can be a good tool for your producers if they have nothing else. Very interesting. If you need to know more, I know I flew through all of this. If you have specific questions, just let me know. And then my information is here. Email me. Our phone is messed up in the lab for some reason. It just goes straight to voicemail. So emailing is the best way to get a hold of me. And I'm done. Sorry, Chelsey.
You're fine. Thanks, Eva. If you want to stop sharing, if anybody has any questions, feel free to pipe in and ask them yourself or put them in the chat.
Anybody? Oh, okay. Yes, I'll send out a copy of my PowerPoint to Chelsey and she'll distribute it. And again, if y'all go through it and you have questions, specific questions, shoot me an email. I'm always around. So.
Okay, if nobody has any questions, there's a poll for you to take real fast on your way out. We will have our last presentation in the series next week, and I'll be talking about nutrition for that one. So thanks, Dr. Wray, for joining us today. We appreciate it.
Thank you. Thanks for having me.